good old sodium potassium pump. And so all of that potassium that had left there, it helps to bring it back in, and it helps to pump all the sodium that came in here, okay, back out, so that you get to the resting membrane potential. Again, how many sodium does it pump in or out, and how many potassium does it pump in or out? 3 what? 3 sodium what? Okay, so the potassium pump pumps 3 sodium out and 2 potassium in. That pump right there helps to bring this charge back to resting membrane potential. Okay, and so that's how, in a nutshell, that's how an electrical impulse is sent. What may be a reason, again, let's check your understanding, what may be a reason why the electrical impulse just stops here at the axon hillock? What might be a reason that it's not sent to the next neuron? Any closer to 0 than -55. Okay. Michelle said it doesn't go any closer to 0 than -55. It stops here. In other words, the signal's not strong enough. It's not strong enough to do this. It might stop at -50. It might stop at -60. Might stop at -56. It has to reach -55. In other words, it has to become more positive by that much. If it doesn't reach threshold, then it stops. If it reaches threshold, then the signal continues. It doesn't necessarily signal any stronger, it just happens or it doesn't. Okay? So it doesn't degrade, it just happens or it doesn't. So all of this, one signal happens between 1 millisecond and 4 milliseconds. That's how all of this happens. 1 millisecond, thousandths of a second to 4 thousandths of a second. So, and that's just remarkable in itself. Now, why might one signal happen faster than another? Okay, well, there are 2 factors involved for how quickly they happen. In other words, whether this happens in 1 millisecond or 4 milliseconds. Okay. Okay, how do I have this? Say, okay, yeah, speed. Speed of an impulse or an action potential, and don't get the term action potential confused with membrane potential. Okay, membrane potential is the differences of charges from inside to outside. The membrane potential of a non-signaling neuron is -70 millivolts, also called the resting membrane potential. And action potential is the actual electrical impulse. Okay, it's like this signal. The speed depends on 2 things: the diameter of the axon. A larger diameter, in other words, we have something like this. Larger diameter increases the speed. Larger diameter increases the speed. Now, why would that be? Okay, so here you've got to just kind of visualize. Let me draw like this. Let me draw one that's small. Okay. Here's a large diameter axon. Here is not as large of a diameter. You have to remember that a neuron is a cell. So it has cytoplasm, there are organelles on the inside, there are different kinds of proteins on the inside, there are other ions on the inside. Okay, so I'm gonna draw, what I'm drawing here along the axon, let me just draw it like this. What I'm drawing are voltage gated ion channels. Okay, same thing here. Voltage gated ion channels. Okay. Now, if an electrical impulse depends on ions flowing in through those channels and out of those channels, which of these two examples, large, small diameter, would decrease the amount of obstacles, for lack of a better word, that those ions would encounter when they flowed in? The larger one, because there's more space for all of those molecules that are inside. Here, you have cytoplasm, you have organelles, okay, whatever proteins on the inside. Look, if the ions flowed in, they would encounter something, and that might be an obstacle. Maybe the obstacle wouldn't prevent them from flowing out, okay? For whatever, to make it simple, larger diameter ions prevent less obstacles, the ions encounter fewer obstacles, I should say, not less. Okay, also, what's going to increase the speed is insulation on the axon. What might that insulation on the axon be? What might be wrapped around the axon? It's fatty. It's lots of lipid. What insulates an axon sometimes? What's that material called? Myelin. Okay, so if the axon is myelinated, if it's insulated, it's going to actually allow the electrical impulse to travel faster. Okay, so myelin, insulation, increases the speed. So two factors. Increase the speed of a signal. Larger diameter, myelination. What is myelin composed of? Lipids and proteins. In what ratio? What percentage? 80 to 20. Okay. And you think about myelin. Okay, let's go back, see what you recall. What neuroglial cell in the central nervous system myelinates axons? Yes, I think I heard oligodendrocytes. Okay, that's in the central nervous system. What about the peripheral nervous system? Schwann cells. Schwann cells. Okay, so both of those oligodendrocytes actually kind of look like this, kind of like a little octopus. Okay, and it's their little process. Okay, this is a cell. It has a nucleus. That's what the little dot is. That little process, that little arm, it wraps itself around sections of the axon multiple times. Sometimes 20 times, sometimes 300 times. Schwann cells are a little bit different in that the whole cell wraps around. So the whole cell wraps itself around, kind of the nucleus is kind of pushed to the surface or to the perimeter. Okay?